Hello everyone, I hope you are all doing well. So today I decided to do this next chapter or this new chapter in our channel where I will share with you my daily life where I'm learning bioinformatics, some courses, etc. So why don't you learn with me? Okay, we will learn together, we will understand together all these terms. Uh, we can say all those courses, this is the first time where I will see them, where I will learn them. So we will learn together and I will try to explain as much as possible and as much as I can so everyone will understand. Maybe these videos will be a little bit long, maybe 1 hour and a half, but trust me guys, you will learn a lot of things and they will find these videos very helpful. Okay, today we will start with this course here. I just started it yesterday and I uh, learned it around 1 hour, and then yesterday night when I was going to sleep, I just was thinking and then this idea pops to my mind. Why don't you share these lessons with your people in your channel, since you have a YouTube channel where you teach bioinformatics? Let's do some videos about these courses. Okay, guys? So let's start. So this is, as I already mentioned, this is the first course. It's called Bioinformatics for the Terrified. But hold on, guys. I don't understand what does the terrified mean, so I will just go here and then ask ChatGPT, what does the terrified mean? I'm learning, guys. We'll learn together. Terrified mean extremely afraid, very, very scared. Oh, this is very interesting. Why does? Uh-huh. So, bioinformatics for the terrified. I don't really understand what they mean 100%, but yeah, let's start. An introduction to the science of bioinformatics. What you should know, guys, is that this course is for beginners, is for people who almost don't understand nothing about bioinformatics. So it doesn't matter to have background knowledge in biology or in informatics to understand these things. Okay, so just watch. What is bioinformatics and where does it fit with bench-based life science research? Okay, this is a question. Find out more about bioinformatics tools and resources uh, that are available and how you can start to apply them in your research. Okay, let's start with the course overview. Who is this course for? Here you will find that this is not for advanced people. This course is aimed at experimental or bench-based researchers working in the molecular biosciences who have little or no previous experience, you see, no previous experience of using bioinformatics databases or tools. It will provide a broad overview of bioinformatics as a subject and the resources available publicly for researchers to access and use. Okay, all understood. And undergraduate and undergraduate knowledge. What does this mean? Undergraduate knowledge. So, guys, here is a trick. If you want to learn any language, for example, for me, English is not my native or my first language, So I'm learning English, but the way I'm trying to learn it is that I'm not trying to do some translation in my first language. So no, when I search about a word that I don't understand or anything I don't understand, I search it in English. Okay, so here's a trick. If you want to learn any language, learn it by the same language. Don't use other languages. Because let me give you an example. If you are maybe Arabic, okay, you speak Arabic and you want to learn English. The problem most people do is that they learn English using Arabic. So they do a lot of translations. When they come to daily life, when they want to speak English, maybe with native speaker or just other person who speaks English, when someone asks them a question, they have the answer in Arabic and then they try to translate it in English. So this take maybe 5 seconds for each one to respond. So if you ask him a question, maybe he take 5 6 seconds so he can reply to you. So this is so bad. You should think in the language you want to learn. So the question was, what does undergraduate knowledge mean? Here we have two responses. Let's see only the first one. Undergraduate knowledge mean the level of knowledge that students are accepted to learn while studying for a bachelor's degree, undergraduate degree. It's a university. So undergraduate, bachelor's students before a master knowledge. Okay. Undergraduate knowledge means the level of knowledge that students are accepted to learn while studying for bachelor's degree. Okay. Undergraduate knowledge is the knowledge people who don't have mastery yet should know. Okay, all understood. This is so annoying. So, an undergraduate knowledge of a subject related to the life sciences would be an advantage. Okay, this online tutorial is part of the introductory bioinformatics pathway, a curated set of EMBL-EBI online course. Let's see what's this. They said here, this curated collection brings together a number of online tutorials and recorded webinars to provide an introduction to bioinformatics, a brief tour of the resources available from EMBL, EMBI, and more details about some of those resources including Ensemble, UniProt, and Expression Atlas. What is this course for? This collection is for anyone with an interest in learning about bioinformatics and the EMBI resources to work through at their own pace. So what does this mean? EMBL, EMBE. European Molecular Biology Laboratory and European Bioinformatics Institute. It is one of the world's most important bioinformatics researchers institutes. Okay. This is where the course is from. I didn't realize it. Is a research institute local in Hankston near Cambridge, England. It's part of the European Molecular Biology Laboratory. Good. Its mission is store biological data, develop bioinformatics databases and software, conduct cutting-edge research, train scientists worldwide. What does EMBL-EBI provide? if you study bioinformatics, you'll use EMBL, EMBE resources almost every day. Okay, it's like a source of data related to bioinformatics and these stuff, as you can see. Okay, sounds interesting. Here we have some databases tools such as UniProt. Its purpose is protein sequences and functions. Okay. Now I understand. Ensemble genome information, interpro protein families and domains, PDBE, 3D protein structure, ORI express gene expression data, MGDNFI, microbiome datasets. Good. These are some databases where you can find, as I already mentioned, a lot of data that you maybe need to work on to do some researchers, exercises, etc. Okay, sounds interesting. Here's an example. Imagine you've discovered a new protein. You want to know what is its function. Is it found in humans? Does it exist in bacteria? Does it have a known 3D structure? You would use EMBL-EBI databases to answer these questions. Okay. It's all clear right now. Good. Let's go back. Let's start again. Which provides an introduction to bioinformatics, a brief tour of the resources available from EMBL-EBI. Now when I read this, I understand what does it mean. Good. This is why, guys, you should always search about anything you don't understand. Okay, let me my coffee starts to get cold. Okay, and more details about some of those resources, including Ensemble, UniProt, all those are datasets. Okay, Expression Atlas. Why we call it Atlas, Expression Atlas? I'm in fact very interested in this name. Why we use, say, Atlas, and because in this area where I live, there's a place called Atlas. I don't know if it's related to this or not. That's a great question. The word atlas in Expression Atlas does not refer to maps of countries. Instead, it has a similar idea. What is an atlas? An atlas is traditionally a collection of maps. For example, a word atlas contains maps of all countries. Anatomy atlas contains details maps of the human body. The key idea is an atlas helps you find information by showing where things are. So what is Expression Atlas? Expression Atlas is a database from EMBL-EBI that acts like a map of gene expression. Instead of showing countries, it shows which genes are active, in which tissues, in which organs, under which conditions, in which species. Example, imagine you're interested in the gene TP 53, okay? Expression Atlas can tell you things like tissue brain as yes. Why Atlas? Because it maps gene expression across the body. Okay. So Atlas, we use it when we have a map there. Word Atlas, map countries around the world. Expression Atlas, map where genes are expressed in different tissues. Okay, so there is no relation to what I was thinking of. Okay, good. Please note that we do not provide certificates. Okay, this course we have do not provide certificates. Okay, doesn't matter. For completing our on-demand training courses. However, if you have an account with us, you can easily track your progress. All the courses you undertake will be listed on my learning page. So guys, what does this tell us is only this course do not gives us some certificates, so it doesn't matter even if you have courses that don't gives you certificates. The most important thing is to understand the knowledge exists in these courses, and you can apply it in your work in the future. It doesn't matter to have real certificates that tells people this guy complete this certificate, okay, or this course. Sometimes it matters, but the most important thing is what is in your mind, okay? Because a lot of people have certificates, but they don't understand a lot of things, okay? They have certificates, but if you tell them to explain to you what they learn or to do a project related to that, they don't know. Basically, they don't. They just want to get the certificate. But if you learn to learn, this is another thing. What will I achieve? So if you learn this course, what you will achieve by the end of this of the course, you will be able to access the role of bioinformatics in molecular science. Great. Describe the key features of primary and secondary databases. List strategies of describing data consistently. Okay. uh, Identify some of the different types of data analysis that can be applied to solving biological problems. There is a lot of data, because that's it. Bioinformatics is simply, you should be a data scientist. Data is very important here. So let's enter the course. Start the course. Let's go. Good. So here is the course overview. Okay, what is bioinformatics? Who is bioinformatics for? The role of public databases, etc. Okay, so let's start with the first one. What is bioinformatics? Put simply, bioinformatics is the science of storing, retrieving, and analyzing large amounts of biological informations. It's all about data, as I already mentioned. It is a highly interdisciplinary field involving many different types of specialists. That's true, including biologists, molecular life scientists, computer scientists, and mathematicians. True, mathematicians also have a big value here. The term bioinformatics was coined by Pauline Hugwig. I don't know how to pronounce this name, man. And Ben Hesper. So, guys, if you don't know how to pronounce name, you can ask ChatGPT. Yeah. How can I pronounce this name? It's all easy, man. Pauline Wigger. Maybe. Let's hear it.
Pauline Hogaveth.
Hogaba. That's more harder.
Pauline Hogaveth.
Pauline Hogaba. Maybe. And Ben Hesper to describe the study of informatic processes and biotech system. What is biotech? Biotech, maybe the combination of biology and technology. Let's see. Biotics means living or related to living organisms. The word comes from life, related to. So biotic, anything that is alive or was once alive. Good vocabulary. I find it very helpful. In biotech systems, okay? Biotech systems. So any organism that have been live, we call it biotech. And it found early use when the first biological sequence data began to be shared. Whilst the initial analysis methods are still fundamental many large-scale experiments in the molecular life sciences. Nowadays, bioinformatics is considered to be a much broader discipline. What is broader? Broader discipline. Broad, something broad. It means, for example, if I'm from Morocco, we can call Europe, UK abroad. It can mean wider, but it's also often used to mean more general, covering more topics or including more things. Okay. So bioinformatics is not only about studying biotech things, but it starts to be much broader, so much broader disciplines. So there is a lot of, we include a lot of other disciplines. Okay. Encompassing modeling and image analysis in addition to the classical methods used for comparison of linear sequences or three-dimensional structures. So it's get improving by the time. Genes, genomes, and variation, cross-domain resources, what's this? Gene, protein, and metabolite expression, protein, sequences, family and motifs, molecular structures, chemical biology, and there is the system. You don't actually understand what does this mean, what this refers to. Let's share it with GPT. What does this mean or refers to? There's improvement here. Development, we started from here to the systems. Maybe systems is more complex. I'm not pretty sure. Let's see, this image is not showing a biological progress. Okay, I'm wrong. Instead, it's showing the main categories of biological data and research that organization like EMBL-EBI works with. Think of it as a map of the bioinformatics world. Let's go through each section. Gene, genomes, and the variation. This is the DNA level. It includes genes, whole genomes, DNA mutations, variation, SNAPs, genome sequencing. Example questions: what genes does a human have? What mutation causes a disease? How is the human genome different from the chimpanzee genome? Okay. So the first one we have here, study where we have they are related to genes, genomes, and variation. For the second one, we have gene, protein, and metabolic expression. This is about what is active. A gene can exist in every cell, but it is not always turned on. True. This category studies gene expression, protein expression, metabolic levels. Example questions: which gene are active in liver cells? Okay, this is where databases like Expression Atlas belong. And the gene, protein, and metabolite expression. This is very interesting. The third one is protein sequences, families, and motifs. After DNA comes protein, scientists study. After DNA comes protein. Okay, because there's an evolution. We have DNA, RNA, and then gives us proteins. So after DNA comes protein. Scientists study protein sequences, protein families, conserved motifs, protein function. Example question: does this unknown protein belong to a known protein family? This is where databases like UniProt are important. Okay, it's like we have a lot of sections, and each section have its own database, like UniProt, like Expression Atlas, etc. The third one, we have molecular structures. Proteins have 3D shapes. True. This field studies protein structures, DNA structures, RNA structures. Example: imagine studying the 3D structure of hemoglobin. Okay, this is the one we have in blood. This is where databases like PDBE comes in. Okay, come in. Here is another database. The one number 5 we have is chemical biology. This connects biology with chemistry. Chemical biology. Yeah, just from the name chemical biology, this connects biology with chemistry. Examples we have drugs, small molecules, chemical compounds, drug targets interactions. Questions like which molecule binds to this protein. The last one we have is systems. This is the broadest level. Instead of studying one gene, scientists study entire biological systems. Examples: metabolic pathways, gene regulatory networks, cell signals, whole organisms, ecosystems. Question: how do thousands of genes, holy shit, how do thousands of genes work together? Okay, this is very interesting. What is the curved line? Okay, there is a curved line here. Here you can see it. Okay, the curved line says cross-domain resources. This means there are databases and tools that work across all these categories. This means there are databases and tools that work across all these categories, yeah, connecting DNA, proteins, structures, chemical and biological systems. So it means there is a relation between all those categories, okay? And mhm, that's normal. How we see it, that's makes sense. It makes sense. It makes sense. Good. Wait, is there a human and plants? The picture shows that these resources are not only for humans. Bioinformatics study. So this is a good point. A lot of people, when we talk about bioinformatics, they imagine we study only humans, but this is not true. In bioinformatics, we can study humans, plants, bacteria, animals, fungi, microorganisms, etc. The same databases can often be used for many species. Okay, this is a good question. The same databases. But why we can use the same databases? This is a good question. Because we are different. The human body is very different from bacteria body. But why we can use the same databases? Why the same databases can be often used for many species? Excellent question. The reason is that all living organisms share the same fundamental biology. Humans, plants, bacteria, fungi, and animals all have DNA, genes, RNA, proteins, cells, the genetic code. Because these biological concepts are shared, the same type of databases can store information for many different species. Okay, so we have the same fundamentals. This is why. Same fundamental biology. Good. Good to know. Guys, we will do stop here, okay? Pause, and then we will continue. So let's restart again. We finished this one. This is the description of the figure one, a broad overview of the different types of data that fall within the scope of bioinformatics. Traditionally, bioinformatics was used to describe the science of storing and analyzing biomolecular sequence data, but the term is now used much more broadly in encompassing computational structural biology, chemical biology, and systems biology, both data integration and the modeling of system. And here we have a good distinction that we should know from medical informatics, because there's a big difference between bioinformatics and medical informatics. Bioinformatics is distinct from medical informatics, the interdisciplinary study of the design, development, adoption, and application of IT-based innovation in healthcare. This is medical informatics. Services delivery, management, and planning. Somewhere in between the two disciplines lies biomedical informatics, the interdisciplinary field that studies and pursues the effective uses of biomedical data, information and knowledge for scientific inquiry, problem solving, and decision making, motivated by efforts to improve human health. Good. Projects such as 100,000 Genomes Project, for which the requirement and sequencing phases run from 2012 to 2018, with all analysis completed by 2021, are bridging the gaps between these disciplines. So this one is very popular, 100,000 genomes. It exists in the UK, in England. Here it is. You can check it later, guys, if you want to understand how does it work. Okay, or you basically can ask ChatGPT about it. But on the whole, bioinformatics deal with research data and uses it for research purposes. Medical informatics deals with data from individual patients for the purposes of clinical management, diagnosis, treatment, or prevention. What is prevention? Hold on. Prevention. Prevention means stopping something bad from happening before it happens. Okay, prevention. It's like you prevent, taking action to avoid a problem. For example, you realize that this guy will be affected by X diseases that will kill him, maybe. So you prevent, and then you stop the issue before it comes to real life. And biomedical informatics attempts to bridge these two extremes. Good. So I think the first chapter, what is bioinformatics, we understand it well. It's not very complicated. It's easy. Now, let's talk about who is bioinformatics for. Who is bioinformatics for? We have here a figure, Leon, postdoctor. Postdoctor is a job when someone have or had a PhD in X field, they can work as a postdoc after that. Okay, you can search about it. What is postdoc job? A postdoc job, short for postdoctoral researcher, is a researcher position that someone takes after earning a PhD. The purpose of a postdoc is to gain more research experience before becoming an independent scientist or professor. Okay, so here is the career path: bachelor's degree, then a master degree, PhD, and here is the postdoc. And then finally research scientist or professor or industry scientist. So good. The goal? To understand what makes a normally harmless bacterium pathogenic in the lungs of people with cystic fibrosis. Here we should distinguish what is the difference between harmless and harmful. Okay, difference between harmless and harmful. The word harmless and harmful are opposites. What's wrong with the Wi-Fi? Okay, harmless, not causing harm or damage. Okay, harmful, causing harm and damage. This is the difference. They are opposite. Harmless. Harmful, something that is bad for you or can cause injury, disease, or damage, such as smoking, too much sun exposure. Some bacteria are harmful as well. So, doesn't cause. Harmless is the opposite. It's something that doesn't cause harm. Example, most butterflies are harmless. The snake is harmless to humans. The side effects are harmless of certain medicines, etc. Okay, good. So the tasks, I'm using a combination of transcriptomics, proteomics, and metabolomics to understand these pathogenic changes better. Okay, now we are just reading the figure. After we will understand why we have it. Okay, so this is the goal. This is the task he will use. So transcriptomics is basically the study of RNA. Proteomics is the study of proteins. Metabolomics. Okay, let's search about them. Why don't we search about them? Here we can do a good thing. Can do like this. This. Good. So we can ask a good question here. What is transcriptomics? As I already mentioned, it's simply the study of RNA. Transcriptomics is the study of all RNA molecules, transcripts, produced by a cell, tissue, or organism at a specific time. Let's break the word down. Transcript, an RNA molecule copied from DNA. Omics stands for the study of all of something. Okay, transcript is simply an RNA molecule copied from DNA. Omics stands for the study of all genomics. So transcriptomics is the study of all RNA transcripts in a cell or organism. This is transcriptomics. Proteomics is the study of proteins, the structure of it, etc. So proteomics is the study of all the proteins produced by a cell, tissue, or organism. This is the same thing. Protein, the molecules that perform most of the work on cells or in cells. Omics is simply the study of all of something. Good. So proteomics is the study of the complete set of proteins. Good. Now it's still only one thing, which is metabolomics. What about metabolomics? In fact, I don't know what does this mean. We'll discover together. Metabolomics is the study of all the small molecules in a cell, tissue, or organism. Okay. Let's break the word down. Metabolite, a small molecule involved in metabolic, example glucose, amino acids, or ATP. Omics, the study of all of something. So metabolomics is the study of the complete set of metabolites. Good. Here we have Barand. He is a plant geneticist, so he studied the, you can say, genomics related to genetics related to plants. Okay, to identify new crop strain resistant to drought, salt, and fungal diseases. Tasks were doing linkage studies to find out which genes are involved in resistance to different types of stress. We've got genomics and expression QTLS that we need to map onto well characterized plants, etc. We have Ola. She's a clinician scientist. Clinician scientist is simply someone who work in real life, for example, a doctor who talk to patients, have a relation, or talk to patients face to face. At the same time, he's doing researchers. Okay, he's improving the field. Good goal to identify proteomics-based biomarkers in urine for the early detection of bladder cancer. Tasks I do mass spectrometry of samples from patients coming in for biopsies. I found a phosphoprotein that seems to be upregulated in some patients. So that's good. Figure 2, the functional person as illustrated here, whilst not bioinformaticians in the classical sense, all use bioinformatics to enable their research cartoons. Okay. So what does this tell us is all those people work in different field related to biology or healthcare somehow, but they all use bioinformaticians somehow. So this is the idea. The molecular life science have become increasingly data-driven by and relying on data sharing through open access databases. This is as true of the applied sciences as it is of fundamental research. Furthermore, it is not necessary to be a bioinformatician to make use of bioinformatics databases, methods, and tools. However, as the generation of large datasets becomes more and more central to biomedical research, it's becoming increasingly necessary for every molecular life scientist to understand what can, and importantly, what cannot be achieved using bioinformatics, and to be able to work with bioinformatics experts to design, analyze, and interpret their experiments. So good. I think, guys, I should stop here, and in the next video we will do the others and explain the others and discover the others together, because the microphone is gonna be off right now. This is why I just speed up. So guys, thank you guys for your attention, and see you soon with the next video. Thank you.